So thank you very much, Dr. Hagiwara, for kind introduction. And I'm Atsushi Miyawaki from RIKEN BS and RAP. And I'm very honored to be here to give a talk to you with this title, Cruising Inside Cells. But let me extend the congratulations to Dr. Honjo. And so today, my talk will be the interplay between the light and life. Okay. So what is light? So light is electromagnetic waves, and life sciences always try to make the best use of light for many different purposes. Okay. For example, we use X-ray beam for crystallography and infrared light for vibrational spectroscopy. Okay. But the most biologists, we use visible light. And I'm going to use this term very often today, chromophore. So it's a structure unit that absorbs visible light at a certain wavelength. Okay. And it is responsible for color. Okay. And in most cases, chromophore has a pi conjugation system, or conjugated double bonded structure. So as you can see here in the beta carotene structure, so single and the double bonds appear alternately, and so that pi electrons can be delocalized over the compound. So that's very important for interaction with visible light. Yeah. So now this slide shows 4 chemical compounds, and all these compounds have pi conjugation system. So they are all very much colored. Okay. So however, so the top 2, so the phenolphthalein and malachite green are not fluorescent. Okay. So after light absorption, so the molecule in the excited state goes down to the ground state, so through a non-radiated process, okay, no emission of light. Okay. And by contrast, so the fluorescent and rhodamine are highly fluorescent. Okay. So they can emit light in their relaxation pathway. So they can go through a radiated process. So what's the difference between the two groups? So the difference is here or here. Okay. So in these highly fluorescent compounds, so 2 benzene rings are breached through an oxygen atom. Therefore, so these parts can take a rigid planar structure. Okay. So the planar rigidity, so in many cases, favors fluorescence, so empirically speaking. Okay. So now let me introduce to you E. coli GFP, so the most famous fluorescent protein. And as you may know, long time ago, in 1962, Dr. Osamu Shimomura discovered purified protein from the light-emitting organ of the jellyfish, and 30 years later, in 92, its cDNA was cloned. Okay. And amazingly, so the protein can be spontaneously fluorescent, and it doesn't require any cofactors or small chemical compounds to be fluorescent or to make a chromophore. And the heterologous expression of GFP was achieved by Marty Sharafi at Columbia. Okay. And let's look at the primary structure of E. coli GFP, just 238 amino acid, the peptide. And there is no chromophore in this peptide. But from these 3 amino acids, serine, tyrosine, glycine, so during the folding into a protein, so 3 reactions occur spontaneously. And 3 reactions. Okay, so cyclization, dehydration, and oxidation reactions, so to make a pi conjugation structure. So this is GP's chromophore, so that absorbs blue light. And you may wonder, so how is the fluorescence? Okay. So the answer is here. So the crystal structure of E. coli GFP and the beta bottle, and the chromophore is buried inside the beta bottle. And with some packing, so the chromophore can take a rigid planar structure. Okay. So that explains why GFP is highly fluorescent. Okay. And also mutagenesis studies have produced several GFP mutants of different colors, and compared to wild type, blue, cyan, green, and yellow. And this was done by Dr. Roger Chen at UC San Diego. And as you know, these 3 people won the Nobel Prize Chemistry in 2008. Okay. And I had a chance to stay in Roger's lab from 95 to 98. And during my stay, I used CFP and YFP so as the donor acceptor for FRET. And I don't go into FRET today, but I created so the genetically encoded indicator for calcium ion. And I named the protein chameleon, so after a reptile animal chameleon. Okay. And the chameleon is the first GFP-based or FRET-based indicator. So now this movie shows developing zebrafish embryo, and the transparency and also external development can provide very good access to almost every embryonic stage. Okay, so we made a transgenic zebrafish line producing chameleon to look at the calcium dynamics. And after fertilization, so there got two big spikes, and followed by some latent time. Okay. And next calcium rise occurred here to initiate so the cleavage period. Okay. Okay. So in this during this cleavage period, so the calcium transient are mostly linked to cell division, and about 10 minutes before, so that cell cleavage, so calcium concentrations increase, so to activate many calcium-dependent machineries. Okay. And side view. Okay. And this cleavage period is followed by brastral period, and the calcium up and down is very localized to the surface of the embryo. Okay. So now I'd like to change the animal from fish to mice, and in the mouse brain, cerebral cortex, we genetically placed chameleon in the excitatory neurons. Okay. And we shine the mouse head, so with the excitation light, and to look at the calcium readout. Okay. So these calcium signals are coming from the entire cerebral cortex, and these signals reflect the spontaneous neuronal activities. And those activities are composed of multiple oscillations with different frequencies. Okay. And the dominant oscillation occurred at 1 Hz, and the activities were always symmetrical. Okay. And the mouse was half awake. So when we gave a visual stimulation, so we saw evoked response here in the visual cortex in the back. Okay. So this is exactly what we are doing at BSI, Brain Science Institute, RIKEN. So I told you about, so they call it GFP and its mutant. And not only the jellyfish, but some other cnidarian animals, so especially corals, so can produce quite similar fluorescent proteins. Okay. So I'm focusing on corals. Okay. And since 1999, we occasionally went to the Okinawa island. And these small islands do not suffer from coral bleaching yet. So we dived into the water and collect these cnidarian animals. And from those animals, so we have cloned quite a few, many new fluorescent proteins. And I think it's very interesting or important to think about why corals produce those proteins, fluorescent proteins. Okay. So then I have to tell you about the dinoflagellate. Okay. So the dinoflagellate are unicellular algae that are ubiquitously abundant in aquatic environment. Okay. And some of them form symbiotic relationships with corals. And the dinoflagellate carry out photosynthesis and give the product, the oxygen and the nutriment to coral animals. However, dinoflagellate are very sensitive to UV light, ultraviolet light. Okay. So therefore, corals produce fluorescent proteins and protect the dinoflagellate from UV. So by using fluorescent proteins as sunscreen. Okay. And the corals and the algae, dinoflagellate, can benefit from each other. So this is symbiosis. And coral bleaching should involve the dissociation of coral-algal symbiosis. The bottom line is the coral-derived fluorescent proteins have some connection with dinoflagellate. Okay. So from now on, I'd like to discuss some unique features found in coral-derived fluorescent proteins. And the first is conversion. And a very long time ago, we went to a pet shop in the downtown of Tokyo and to come across very beautiful, colorful, stony coral, Trochophilia. Okay. And at first glance, I thought we should be able to clone more than one different fluorescent proteins, but I was wrong. And Ryoko Ando in my lab cloned only green-emitting fluorescent proteins. Okay. But one day, she happened to leave the protein sample on her lab bench here, and her lab bench is closed to south-facing windows. And she comes to the laboratory very late. Okay. So the next day, before she showed up, the tube had been exposed to sunlight significantly. And she came and found that the protein sample had turned red. Okay. So this is a serendipitous finding. And so this is for the conversion. And originally the protein emits green fluorescence, but after irradiation of the violet light or ultraviolet light, so it becomes red irreversibly. And we named the protein kaede maple leaf in Japanese. And these three culture neurons expressed kaede protein. So the beginning everywhere green. So it was very difficult to delineate individual neurons. But these two somata were illuminated by violet light. So for 0.5 second and 0.25 second, half a second and a quarter second. So then we enjoyed autumn colors on the petri dish. Okay. And Dr. Hata at Kobe CDB generated kaede transgenic zebrafish, and so he followed the optically labeled the cell populations during the embryogenesis and to perform cell lineage analysis. Okay. And successfully we elucidated the molecular mechanism of the photoconversion. Okay. And this is essence, and the kaede has HYG, okay, histidine, tyrosine, glycine as the three amino acid for chromophore formation. And the histidine has imidazole ring as a side chain. Okay. And again, through the same three reactions, cyclization, dehydration, and oxidation reactions, so green chromophore is made. Okay. And then UV irradiation brings about the change in charge distribution like this, and the cleavage of the peptide. Okay, main chain is cleaved. So this is a beta elimination reaction. The leaving group is a carboxamide, and the proton is removed, and the new double bond is introduced here. So which connect the imidazole ring with the histidine to the original green chromophore. And now the pi conjugation is beautifully expanded. So when we reach this conclusion, we are super excited. Okay. And the photoconversion is irreversible. But the next is photochromism, and it is photo-induced reversible transformation of a molecule between two forms. Okay. And after our paper on kaede came out, Ryoko brought me a new next coral sample. But I was not so happy when I saw it, because very shabby. Okay? So compared to the Trochophilia which produced kaede protein, the new sample was very dirty. Okay? But from this coral, she cloned very bright and green-emitting fluorescent protein, and then she performed in vitro mutagenesis. And after relentless mutagenesis, she made it photochromic. So now we can convert the protein between the dark and bright state, using two different colors, blue and violet, and on and off regulation. And we named the protein DRONPA. So DRON is a mimetic word for disappearance, and the PAS stands for photoactivation. And we prepared the recombinant DRONPA protein to make a rewritable molecular memory system. Okay. So these studies on Drompa protein gave us 2 lessons. Lesson 1, don't judge a coral by its appearance. Okay. And the lesson 2, even if a wild type fluorescent protein is dim or boring, don't throw it away. And we may be able to make it brighter and more interesting and through mutagenesis. And we could go beyond the corals and the jellyfish. Okay. And I'm very happy to say both Kaeda and Drompa made some contributions to the invention of super-resolution light microscopy. As you know, the spatial resolution of the light microscopy is limited by diffraction to about 200 nanometer in the focal plane. Okay? But in order to break the diffraction limit, so recently some new advanced microscopy systems have been developed. Okay? And Stefan Hell and Eric Betsy, they invented the STED microscopy and the PALM respectively, and in their proof of concept experiment, so they used guided Drompa. Okay. And a couple of years ago, we assembled at the Nanyang Tech University, and the conference or symposium featured Dr. Craig Venter, the next speaker too, and I was Bruce Lee, and he was Superman. Okay. All right. Next, I'd like to discuss the bioimaging at the organ and the tissue level and the cell cycle. Okay. So this is a cell cycle of eukaryotic cells, G1, S, D2, and M. And before dividing, to produce 2 daughter cells, a cell is supposed to duplicate the DNA in the S phase. Okay. So we wanted to visualize the transition from G1 to S, G1S transition. Okay. And for that, we used ubiquitin oscillator, the cell cycle dependent proteolysis. And these 2 E3 ligase activities oscillate reciprocally during the cell cycle. And their substrate include CDT1 germinin, and these 2 protein levels also oscillate reciprocally. And we used the ubiquitination domains of these proteins and plus our own green and red proteins to create the cell cycle probe which labels individual nuclei in G1 phase red and those in SG2M phase green. And now G1S transition can be monitored as a color conversion from red to green through yellow. Okay, so that's food. And when food was introduced into HeLa cells, so coming from malignant tumor, we were able to observe the cell cycle progression in real time at single cell level. Okay. But when we did almost the same thing, so using NAMU MG cells, so derived from benign tumor, so we saw very clear contact inhibition. Okay. And upon reaching confluent monolayer, all of the cells became red and stopped their proliferation. So probably G0 arrest. And we introduced a scratch. Okay. And then we saw some of the cells at the edge of the scratch were turning green after some latent time. So they re-enter the cell cycle, and they increased the number to build the gap. So this is a very good wound healing model. Okay. And in a cell transplantation experiment that used nude mice, we could distinguish between benign and malignant properties of the tumors by color. Okay. And we made a transgenic mice. And this is coronal section of the embryo, embryonic day 13. Okay. So here, every cell nucleus shows either green-red fluorescence depending on the cell cycle phase. And in the brain, so neural stem cells had only had a green-red nuclei in the ventriculosome, but the post-mitotic neurons had only red nuclei in the cortical plate. Okay. So the green signal should indicate cell proliferation and the red differentiation. Okay. All right, so the embryonic day 8.5, 9.5, 11.5, So with time, so green to red ratio decreased. Okay. So the earlier, the more proliferation, the later, the more differentiation. Okay. And after birth, so the red signal become predominant. But even in the adult tissue, so we can identify very few green nuclei. And the cells with the green nuclei so correspond to tissue stem cells. Okay. And which can be applied to human iPS cells, and of course zebrafish. And in the zebrafish embryo, we can obtain the cell cycle profile in the 4 dimensions, X, Y, Z, and T time. Okay. So this is a segmentation period, and side view and the head and tail. And again, with time, green to red ratio decreased, but some mitotic organs, including retina and the brain, are very green. Okay. And top view, in addition to 2 eyes, so we observed the differentiating node code. Okay. And the node code is essential for patterning plaques and mesoderm. So you can follow sequential formation of somite here, and plus the differentiation node code. Okay. So the node code is the central region of the body. Okay. And I should say it's amazing so that we can visualize the central domain of the animal in the live state, so from outside. So this is because fish embryos are extremely transparent. Okay? And by contrast, mouse embryos are turbid. Okay? And the mice and the human and many animals are not transparent. Okay? And the visible light scatters very much in the body or inside the tissue. Okay. So now I'd like to touch upon tissue clarification technology a little bit. Okay. So long time ago, we discovered the PBDF membrane for Western blotting became transparent when soaked in formaldehyde. And we knew that the beta bottle structure of fluorescent proteins very tough and tolerant of high concentrations of urea. So these findings was fact. So inspired us to develop tissue clearing reagent, and we made a scale A2, and the formula is very simple: formaldehyde and 10% glycerol and 0.1% detergent. And the scale A2 can clarify the fixed biological sample, and scale A2 can eliminate light scattering. Okay. But it doesn't eliminate light absorption. So scale A2 preserves the color information. Okay. And we already showed many examples of large-scale 3D reconstructions of GFP expressing cells and neurons. And in this experiment, so we appreciate very complex hippocampal formation from different angles. Okay. And I think these tissue clarification technologies should be well combined with, for instance, brain ball technology, which can cause individual neurons to fluoresce in totally 90 distinct colors. Okay. And now, my laboratory belongs to the Japan project, Brain Minds project. Okay. And we are supposed to map neural circuit in the whole brain of rodent and the primate. Okay. And also in my laboratory, we're very much interested in aged brain or aging brains. And for example, Alzheimer's disease is a very famous neurodegenerative disease characterized by amyloid beta, A-beta plaques. Okay. So we are using Alzheimer's disease mouse model and a very aged mouse. And we immunostain A-beta plaques and in the whole brain, and a 9-month-old, so almost nothing. Okay. But at 18-month-old, okay, so we saw so many A-beta plaques scattered throughout the entire cortex. And in addition, we labeled the blood vessels with the red fluorescence, and this is a very comprehensive perspective. So tells us, which tells us how A beta plaques interface with blood vessels. Okay. All right, and I told you about a variety of fluorescent proteins today, and they can be classified into two groups. Echoria GFPs coming from Echoria Victoria, and the GFP-like proteins from other animals, so mostly corals. Okay. But all these proteins share the same basic subunit, okay, beta bottle, and they all form synthesize chromophores from their own peptides. Okay. But in a broad sense, fluorescent proteins should include the proteins that become fluorescent by taking up the natural chromogenic ligand. Okay. So I like to tell you the eel of protein. Okay. And as you know, Japanese eels are very much green fluorescent, okay, and they show catadromous migration. Usually they leave lakes and rivers, but adult eels migrate into the ocean to spawn here. And their larvae are carried by this current and reach Japan at the stage of glass eels. Okay. So fluorescent glass eels, and in the transverse section of a glass eel, we observed very strong green fluorescence in deep muscle layer. Okay. And in 2009, Dr. Hayashi at Kagoshima University reported the presence or existence of the fluorescent protein in eel muscle. Okay. And then we successfully cloned the gene encoding for the green emitting protein, and we named the protein product UNAG. And so the protein belongs to a fatty acid binding protein, FABP family. So FABP is a carrier protein of lipophilic substance. Okay. And amazingly, so the heterologous expression of UNAG in bacteria did not produce any fluorescence. But in mammalian cells, very bright green fluorescence. So we hypothesized that the protein required some factor to fluoresce. And we spent 2 or 3 years to identify the ligand X. And finally we discovered bilirubin is the ligand. Okay. And the mixture of the bilirubin to apo-UNAG, okay, so resulted in immediate development of the green fluorescence. Okay. So now what's bilirubin? So here, it is heme metabolite. So this slide shows heme metabolism. Heme, biliverdin, and bilirubin, and its conjugate. Okay. So I show you the structure. And bilirubin has 4 pyrrole rings, A, B, C, and D. And the rings A and B are pi conjugated. Likewise, C and D. Okay. So these 2 dipyridine absorb blue light. As a result, so bilirubin looks yellow. And we determined atomic structure of horo-UNAG protein complex at 1.2 angstrom resolution. And in the binding pocket, from top, AB and CD, okay, AB and CD, and planar conformation can be verified and from a certain angle. Okay. So again, planar rigidity looks very fluorescent. And bilirubin is lipophilic, it's not water soluble. So in our blood, it's bound to albumin and transported to the liver. And in hepatocyte, it is converted to bilirubin glucuronide. And this water-soluble molecule can be excreted into bile. Okay. And in this metabolic flow, when hemolysis is increased or hepatic function is impaired, bilirubin may accumulate inside the blood, also in the extravascular tissue, and that should cause jaundice. Okay. And very efficient diagnostic method is really required. So that measures serum levels of bilirubin. So there are many significant problems concerning bilirubin measurement in the clinical laboratories worldwide. And so to make a long story short, we generated fluorometric method for bilirubin measurement. So just using our apo-unagi protein expressed in bacteria. Okay. So this is highly sensitive, very sensitive, precise, simple and rapid. And for example, so our method is 10,000 times, 10,000 times more sensitive than the current method. And now so many, more and more pediatricians are approaching us for monitoring the serum levels of bilirubin in infant, especially premature babies. And in those babies, high levels of bilirubin should cause connectors brain damage. Okay. But at the same time, so more and more researchers take interest in bilirubin as a powerful antioxidant. So it may be more than a heme metabolite. In fact, bilirubin is easily oxidized back to biliverdin. Okay, so that suggests it may function as an antioxidant. Okay. So clinical evidence is accumulating that the moderate increase in serum levels of bilirubin is associated with reduced risk of many diseases that involve oxidative stress, such as diabetes and neurodegenerative diseases. Okay. So it may be a new barometer of healthcare. And we really hope that our studies on unagi protein will contribute to the preservation of this biologically mysterious and economically significant and ecologically threatened fish species unagi in the future. Okay. And I'd like to come back to this slide, so the interplay between light and life. And now life is creatures on Earth. Okay. And as you can imagine, so animals, plants, fungi, protists, and even bacteria utilize light very ingeniously. And at the same time, so they are fatally affected by light. And my laboratory is engaged in technological innovation in the bioimaging, so principally using fluorescent proteins, and about the physiological roles of fluorescent proteins in the interplay. So there are many riddles, enigmas. So finally, I'd like to pose 3 questions. Okay. So the first question is this. So what excites E. coli GFP in the jellyfish, E. coli Victoria? And the answer is echolin. Okay. So echolin is labbed by Shimomura. So Dr. Shimomura labbed echolin greatly. And echolin is a calcium-sensitive photoprotein, chemiluminescent protein. And when purified, it emits blue light in the presence of calcium ion. However, in the light-emitting organ, so echolin is tightly accompanied by GFP. And echolin's excited state is very efficiently transferred to GFP. As a result, so the animal emits green light instead of blue. Okay. But nobody can tell exactly why jellyfish emit light and why they shift spectrum from blue to green. Okay. Second question, what excites fluorescent proteins in the corals? Sunlight, probably. And I hope you remember so the coral-algal symbiosis. Okay. And so the fluorescent protein may protect dinoflagellate from UV light, but that's only hypothesis. Okay. Number 3, what excites so unagi in eel muscle? Well, so the moonlight may do it, but we haven't the slightest idea why. And in those, in spite of those enigmas or mysteries, I think we need to respect the glowing eels, corals, jellyfish, plant, or whatever nature. Okay, so the nature still remains the best source of new tools and new technology. Okay. And when Equator GFP was awarded a Nobel Prize in chemistry, I had opportunity to write something, and this is the last paragraph of my article. And so just highlight the last two sentences, and please let me read out. And Dr. Roger Chen, my previous advisor, is in the habit of reminding us that the nature has been kind, and perhaps only a scientist with the respect for nature so would have been permitted to surpass it. So this is take-home message in my talk today. And so I stop there, and thank you very much for your attention.